Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. Samantha Bailey-Bucktrout is the Senior Vice President of Research at Akamis Bio, a company harnessing the power of tumour gene therapy in the fight against cancer. Samantha talked to us about her career in cancer immunotherapy, what Greek mythology has to do with modern-day biotech, and the importance of a collaborative approach to drug development. This week, I am delighted to be joined by Samantha Bailey-Bucktrout of Akamis Bio. Samantha, welcome to the show.
Thank you so much, Tom. I'm really honoured to be invited to this show and discuss my career and also my current role at Akamis Bio.
Absolutely. Well, we're honoured to have you, Samantha. Thanks for coming on. And I wanted to start by talking a bit about the work you're doing now. It's where we typically kick off. And what was interesting when I was looking into the company was that, of course, a lot of cancer companies are taking a cell therapy approach to cancer treatment. You're taking a slightly different angle. I believe that's reflected in the name as well. So it'd be great to hear a bit more about what you're doing and about the approach that Akamis are taking.
Fabulous, yes. So Akamis is a gene therapy company that utilizes a viral vector platform with which to deliver therapeutics. Akamis Bio, interestingly, was taken from the name of one of the warriors that was in the Greek mythology story of the Trojan horse.
Okay.
Yes, so Akamis was a warrior who was within the horse, of course, entered the besieged castle and then destroyed the castle from within. And here within our therapy, which is delivered systemically, which is rather unique across the space that we work in, we are delivering DNA payloads that then get translated within the tumour itself. So this is a tumour-expressed payload. And we can put up to 4 novel protein therapeutics that are expressed within the tumour microenvironment. And when you look at our pipeline, we've been choosing immunotherapeutic proteins. So we're really enhancing immunogenicity within the tumour microenvironment, and then have the ability, of course, to co-opt the immune response and subsequent TME responses, as is our desire based on what we express.
Yeah, I see. Okay. And you're developing both the delivery mechanism and the payload, are you? Or what's the...
Yes, absolutely. Yes. So that's absolutely right. So the delivery mechanism comes from a legacy first generation product that was generated within the laboratory of Len Seymour at Oxford University. And this was part of what is the kind of adenoviral wave of gene delivery tools, of course, of which are COVID vaccines, so that came out of Oxford Uni, and that AstraZeneca then took on along with Biotech to manufacture the platform. So that's one version. Our version is a very unique serotype. So most of the adenoviral vectors that are currently in preclinical clinical development are serotype 5. We're a chimeric, a novel chimeric that comes from serotype 311. And that what was so compelling from a novel therapeutic perspective, and what Len Seymour and his group did was take quite a large pool of viruses and do a directed evolution. Right. So it's kind of like the generation 1B of viruses that has generated a virus that replicates only in malignant cells. So malignant epithelial cells support the replication of the virus, and not in any human cells that have been tested. But then also importantly was this virus can infect these tumor cells in the presence of whole blood. So this screen enabled kind of selecting for low immunogenicity within the patient population. So we have the ability to IV deliver. And this is what sets apart from many of certainly within the adenoviral serotype 5 space, because of strong pre-existing immunity. Adenovirus 5 is a virus that infects many children, and it gives you common cold symptoms. It's kind of a cold virus, hence has been used for therapeutics because it doesn't form a really serious pathogenic safety risk. So ours, our platform doesn't have this pre-existing immunity. And unlike a lot of other viruses that have a directed engineering approach that then supports tumor-specific replication or even entry, because it's been selected in this way, when you look at the gene deletions and the genetic map of this virus and the mechanisms of selectivity, it's very broad and hits a lot of kind of known pathways, such as metabolism, such as DNA sensing pathways that tumors tend to downregulate. So we've got a multifactorial tumor-specific mechanism all built into this. And it was the virus that determined it, not us. So sometimes biology is smarter than us, unfortunately, unsurprisingly.
You know, I did one of the very early interviews I did on this podcast was a company that were doing work in immunology. I can't remember the exact detail, but the CEO of that company said something that was really interesting, was that the immune system is so good that it's amazing that we're even alive with the amount of things that are trying to kill us. That's how good the immune system is. Like, we shouldn't exist because of so many things that are trying to kill us. So why reinvent that?
Absolutely. Yes. To quote Jurassic Park, nature will find a way.
Exactly. And so, correct me if I'm wrong on this, but from what you're saying then, there's also an advantage in that the payload itself actually is what destroys the tumor, rather than having to rely on there being strength left in the immune system in the patient.
Yes, well, it depends which payload that you express, of course, Tom, on what we're relying on the body to provide. So, for example, our clinical candidates, so we're a clinical stage company. We've done a lot of work with our empty vector first to establish the tolerability profile, to have a very large kind of transmed and safety package, which really supports the mechanism of the platform, that it's tumor-specific replication and transgene delivery. So that's been very important, a very important stepping stone within our phase 1A studies, not only for dose finding, of course, and the safety, but really proof of concept of that our virus is going to be able to be delivered systemically, number one, and then number two, that it's safe and that transgene expression isn't all over the body, because of course we have a lot of epithelia throughout the body. And these have been very carefully done, and we're pleased to be moving forward to a phase 1B study now, where we're really looking at efficacy.
Fantastic.
So within the clinical, our lead clinical candidate, it expresses a full-length anti-CD40 agonist antibody. So this is what, from an immunological perspective, this is right at the get-go of an immune response. So this is, if the immune response is a car, and and we're wanting to go from A to B, this is basically us putting the key in the ignition and turning on the starter motor. CD40 kicks off and is very potent at kicking off an immune response. So for that, we are relying on some things being there to be able to kick an immune response off against the tumor. That is dead tumor cells, of which the immune system can pick up, present, and recognize. So in that concept, we're relying on things, but there are other scenarios which we've recently published in collaboration, and we're continuing to work with a new collaboration and you mentioned CAR T. So for example, our vector program and our preclinical assets combine really well with CAR T.
Yes.
So taking a therapeutic that negates the need for a very robust immune response in patients, because you're adding the anti-tumor effector cell, and our virus can combine really well, not only enabling a more potent and durable CAR T response, but we can redirect CAR Ts that have already been approved in the clinic into new indications and new patient populations, based on what we express in the tumor.
Fantastic, which should hopefully make you a shorter pathway to doing that, right?
Yes, absolutely. Yes.
Yeah. It's fascinating. And so, yeah, I suppose the amount of applications you could have just within oncology with this sort of therapy are vast. Are there particular areas that you guys are focusing on to start with?
Yes, absolutely. So antibodies, as I mentioned, we have a full-length antibody. And actually a full-length antibody is quite an enormous molecule and takes up most of the available amount that we have within our available to play with from a DNA payload perspective. But now what we've been able to do in the preclinical research team is express antibody fragments. So of course, everyone's heard, I'm sure, of bispecific SCFVs. So these are single chain fragments with the light and heavy chain, but you can make them multi-specific.
Yes.
T-cell engagers are a great example here. You can do tri-specific. You can follow that field if there's a need within the tumor microenvironment, but also nanobody fragments, which these are have the advantage of being, some of the smallest proteins and therefore high penetration. So if not only are these extremely challenging to manufacture and deliver, and deliver effectively with their poor PK and clearance, getting them, imagine this, we don't have to deal with manufacturing because the tumor cells are the drug factory, and then they're in the tumor microenvironment already. So it's just how do you effectively then engage the effector arm of that, whether that's a T-cell and a T-cell engager or other aspects. But we can do other things like talking about antibodies and other approaches. You can theoretically redirect antibody drug conjugates, for example, which there's been breakthroughs lately, at least from a tolerability of dosing perspective. So again, you've got an anti-tumor antigen specific antibody bringing in a cytotoxic agent. So, a less toxic chemotherapy that works for a single tumor antigen now, a single indication. Again, we can broaden that to other indications because we can express that tumor antigen in any other epithelial tumor type.
I see.
So yes, we've got a very long list on the whiteboards at Akamis Bio of all the things we can do. It's more about how many hours do we have in the day and dollars in the bank.
Absolutely. No, it's very exciting. And your role there, Samantha, so you're the Senior Vice President of Research. So clearly, still very connected to the science of the company and the work you're doing there. But just tell us a bit about where you spend your time, what you focus on, where's your attention taken up these days?
My attention very much these days is, as I've slightly been alluding to, is really thinking about the possibilities of the platform, coming up with priorities there and ensuring that we as stewards of the company and the resources in the company are kind of directing and managing those appropriately because of the large potential of the platform. while also as well, a major part of what we do in research is support our clinical colleagues in the clinical development
Yes.
and the transmed, because we're very passionate here about the whole kind of field of reverse translation.
Mm-hmm.
What's happening in the clinic should very much be informing what the research team is working on next.
Yeah, okay.
Yes, and vice versa. So, you know, not only is there a great potential from the pipeline and the preclinical pipeline that we could develop and deliver, but it's what's relevant for the clinic. What do the patients need?
Mm.
Not only where is there an unmet need, but what patient population is the best fit for our therapeutic.
Yes.
So it's very, of course, I think as we are all in biotech and early stage drug development, we're very aware that the largest hurdle, of course, is hitting your clinical proof of concept
Right.
for your delivery. So when there's therapeutics that have such broad potential, I think it's very important to have a very clear picture
Mm.
of your drug profile and fit that correctly within your patient population.
Yeah, that makes sense. And to be as agile as possible with how that feedback loop works.
Yes, absolutely.
Yeah.
So as much as it's very challenging within a phase 1 patient population, of course, given the
Yeah.
goal of that is safety studies, to really extract relevant transmed data, which is very helpful for the research team,
Mm-hmm.
your proof of mechanism, biomarkers of response or resistance, all of these things. It's still an important place to spend time and resource, especially within, of course, with biotech, where, of course, company lifespans are very much dependent on your success within the near term. So that's another area where we do spend time, and really communicating the expertise within the research department, which has been around for quite a while, actually.
Mm.
Phenomenal expertise across the functional areas within research, and communicating that cross-functionally, internally and externally with collaborators.
Yeah, that makes sense. That makes sense. I could ask you so many more questions about this and how it works and how you're managing to have that reverse translation work and all this kind of thing. But we're mostly here to talk about you, Samantha.
Oh,
So,
yes.
but cancer immunology in general, and cancer immunology in particular, has been a real area of focus for you in your career, hasn't it? And I
Yes.
always like to go back to the beginning and
Mm-hmm.
understand why science, why immunology, why cancer, why this career for you?
Yes. Yes, I think, like many and very ununique to your other interviewees that you've had, and I've really enjoyed, by the way, listening to your podcast. I'm a huge
Oh, thank
fan
you.
and follow along, and I think it's wonderful and insightful, and I think for folk that listen, you feel part of the community, and
Mm.
especially that it's UK based is fantastic.
Yeah.
So I think non-unique, interested in biology from an early age. And actually, I remember in high school, my light bulb moment was a genetic, within hearing about genetics,
Okay.
and just that you have 4 nucleotide bases that are the fundamental roadmap of everything that is a complex
Hmm.
organism like a human. And that just blew my mind.
Right.
And then also it was a female teacher as well, and I've seen throughout my career also, I've been very inspired by strong female thinkers and
Yes.
leaders too. So that was the twofer, if you will. And all the way through, I think when I think about what did I feel like, of course, excitement really drove decisions and next
Hmm.
step decisions, and the long hours working. Um, the long hours working in the lab, and coming through all the challenges that we have in this career.
Yes.
It's this fascination again that there's such a simple very simple mechanism right at the center of a highly complex organism
Yes.
or network or system that creates such, the wonder of what we see today. So, I find immunology all the way back to even like, I went to Aberdeen. So initially I was going to be, of course, a vet. Everyone wants to be a vet.
Right.
And then zoology. And then I went up to Aberdeen for my undergraduate zoology, ecology. I was interested in ecology as well. Again, how can small individuals make up an ecosystem, which then is a completely different beast. And I just love this idea of synergy and cooperation.
Yes.
2 years in, though, I spent a summer up in the Highlands looking through sheep dung at grass species, which kind of made me change my
I can see
career
that.
track.
Yeah.
Ecologists are amazing, but I realized it wasn't for me.
Yes.
And then went back to cell biology. And again, like, even I still find fascinating, I could have spent a whole career just working on cell biology, like how a cell with all its complexities organizes itself and back the whole levels. And actually, that was going to be my choice. I'd heard from my friends that were a couple of years ahead of me at undergrad, don't do immunology. It's very complicated. It's full of acronyms and letters and, it's like a foreign language. And I said, oh, okay, that sounds good. I'll just stick to cell. But thank goodness the honors program became merged. So it was cell immunobiology.
Right.
So that was a very lucky tangent for me. And then I did the immunology course and absolutely fell in love with it. Again, because you've got such a complex system.
Yeah.
And it's one of the only systems in the body that moves around,
Right.
It's not a static organ or cell. So not only is it highly complex with very different cell types that have different functional states and trajectories to get to those, it moves around as well. It's phenomenal. So what are the rules that keeps this whole system in check?
Yes.
As you mentioned at the beginning, Tom, we're constantly fighting all of these, external infectious diseases and other things, but that our immune system stays in check. And autoimmune disease is devastating, but it's still relatively, not a massive impact on human health. It's a phenomenal thing. And I actually spent the first 15 years of my career in academia studying immune regulation.
Right.
Through different cell types within the field of autoimmunity. So here's the problem. We have an immune system that instead of attacking foreign agents is attacking our body.
Yes.
What's gone wrong there? And then how do you fix it? So that's what I did. So anyway, did my undergrad. I had no aspirations. I had no academic aspirations. I came from I was the first person in my family to go onto a degree.
Right.
And had huge support from my family, in particular, my grandmother. And I think that also was very helpful for me, because if there was no inbuilt expectations, then you're not going to, what's the risk?
Yeah.
Let's just do it.
True.
I did internships. I was amazed that you could get funding. You could come up with an idea, write a 2-page grant and get funding.
Yes.
And spend the summer in the lab rather than waiting in a coffee shop, being a waitress in a coffee shop. So this was kind of the track of what I did.
Okay.
Interesting projects.
Yes.
Continuing to look, asking the questions of how can these cellular interactions, particular proteins, particular secreted protein messengers, how could they have such dominant impact onto health and disease? And that took me to my PhD was at Edinburgh University. when I started my PhD, dendritic cells, which are, you know, one of the huge control cells of the immune response. This was a burgeoning field.
Right.
They'd been identified and were being studied, mainly generated from mouse bone marrow in the lab.
Yes.
And I found that there was a lab at Edinburgh University that would cannulate sheep lymphatics. So this was a very simple operation, and lymph could be collected from sheep, and about 200 mils of lymph would be collected per day.
Okay.
So one could collect the lymph draining the skin into the draining lymph node. And here were natural bona fide dendritic cells. And
Right.
one could ask vaccine questions. We would use gold bullet vaccines and track which dendritic cell population was carrying the cells, and could do studies on them ex vivo. You know, that was very, it wasn't really challenging the sheep at all. They hang out in their pen and do their thing. So it was a phenomenal model.
Yes.
Very difficult for reagents. That was the challenge.
Yes.
And it was the interesting subject and the next question that kept me going.
Yeah.
I then moved to the US.
Yes.
Met a professor at a conference who was using viruses, interestingly, and vaccines to induce different types of models of multiple sclerosis. And in multiple sclerosis, it was found that you have these waves of T-cell responses. This was the field of epitope spreading. And the question is, how do these new T-cell responses that drive this terrible disease and symptoms of the disease in relapsing remitting MS, how do these get initiated? And dendritic cells were a strong hypothesis, but this hadn't been looked at. So I went to his lab in Chicago, and that's what I looked at. And indeed, within the mouse models, this is what we saw.
Yes.
And then that moved me next to, well, here's a cell that regulates the immune response. What else regulates the immune response? Met another professor who came to visit, who was one of the leaders in T regulatory cell biology down at University of California, San Francisco. That's Jeff Bluestone. And invited me to his group to ask a similar question. How do dendritic cells and Tregs interact? Do they? Looking at different disease models, and was there for around 5 years, looking at the interactions there. And we along in collaboration with 2 other postdocs, so there was 3 of us together, made a discovery around how regulatory T cells actually, under certain inflammatory conditions, can become unstable, lose their identity. And actually, they're like now the bad cops. They actually cause trouble rather than stopping trouble.
I see. Okay.
Yeah. And this was quite a significant breakthrough. And through working in this lab, I really became to value collaboration, close collaboration with people with different expertise and background coming together. Like, we executed on this study like in record time for academia, because we each 3 came with very different ways of thinking and also expertise to apply together.
That's interesting that you learned that early on in your career, because I think sometimes it takes people a little bit of time to understand that. They do their PhD where they have to be the expert in what they're doing.
Yes.
And there's real value in that. But a lot of exciting things happen at the interfaces between people's expertise, right? And it's, you know, it's rare that someone can do it on their own.
100%. And for me, this is, I think, if I was asked what's the number one thing that I've learned through my career, it is, you know, we all can use the word collaboration, and you can see it on mission statements all over our industry, you know, but absolutely, I you know, living that and seeing that those have been the inflection points for me anyway, throughout my career to science that I really believe is impactful. And yes, has been appreciated by the field as impactful. So that's always nice. But
Yes.
living that and really seeing, here I've got a good idea, but it would be really phenomenal if that could be added. This level of expertise, this tool, this way of thinking.
Yeah.
And then also, even if you start that exploration, there's a bunch of things out there that you didn't even know existed that would be better to enhance moving forward, how we move the science forward.
Yes. Yeah, fascinating. And then so up to this point, you've very much been led by a series of scientific questions that have kind of led on to each other. Did that remain to be the case?
Then I think what happened, I did move into industry after 10 years postdocing in the US. It was more, well, I guess it was, but on a much broader sense. It wasn't an individual science immunologically based question. This was the beginning, this was the burgeoning of cancer immunotherapy.
Right, yeah.
Not only do I love the immune system and, you know, everything that it can do, now we're able to use it as a therapeutic.
Yes.
So this was, I had to get after that.
Yeah. I understand.
Yes. And within Jeff's lab was part of a collaboration with industry, within this great incubator that was set up at UCSF, and Pfizer was running that. So I was collaborating with Pfizer scientists. And when I was invited to go join the group, hey, you want to come and do some drug development in cancer immunotherapy targeting T cells? Yeah, sure. That was absolutely.
Yeah.
So that was my move into industry. And it was a phenomenal place because little did I know then, but now I know now how special it was, because this was an R&D unit. It was a biotech in South San Francisco, a Genentech spin out that had been acquired by Pfizer.
Okay.
And Pfizer R&D made the decision to keep the talent, keep the CSO, and let them do their thing. And this was protein therapeutic led. The CSO was a protein guy. And so the team, like, that was the core essential unit of the team, and it was indication agnostic. So then there was groups of biologists like around. So if the center was the protein engineers, the biologists were around saying, hey, what cool proteins have you got? What can we do with them?
Yes.
So it was truly fabulous, and you know, to go in there and learn drug development, and again, collaboration. This was a collaboration overload party, you know.
So you'd clearly already put emphasis on collaboration. Was there more emphasis organizationally within Pfizer on collaboration than you had previously in that case?
I think because this was quite one would think across, of course, across large Pfizer, yes. So as certainly as programs move towards the clinic, yes, absolutely. And you know, one of the things there, of course, you know, within the 5 years I was there, super fun doing the discovery science and learning an awful lot around protein engineering and all of that stuff. The other point that was phenomenal for my career was seeing the drug development path, working with amazing development leaders.
Yes.
And seeing, you know, understanding all of the different careers and the emphasis that they bring. So now it's about collaboration, but not just at the scientific level, 3 immunologists working together. This was collaboration at the, you know, at the functional level.
Right.
And again, with really excellent teams coming together, that's magic.
Yes.
You know, to be part of to be a research lead on an asset and be discussing with the development lead and the translational medicine lead, you know, as well as the clinician, and having everyone there at the table. We had the lawyers there at the table. We had, you know, a little bit of the commercial team. BD was there. So we had a small ecosystem that could all work together.
Yes.
And again, I think that's when value creation was really evident for me. And I really embraced and enjoyed all of the interactions and opportunities to be working with those individuals.
Yeah, that makes sense. That makes sense. And then, so after a period of time at Pfizer, though, you took a step into not back into academia necessarily, but into a different type of organization.
That's right. So, as what happens with, you know, with life cycles of organizations, what was happening was, you know, eventually it ended up programs were being moved and there was things happening. So I was interested in exploring outside opportunities. My lovely professor and mentor, Jeff Bluestone, was president and CEO at a newly started adventure, the Parker Institute for Cancer Immunotherapy. So this is in San Francisco. This has been a very successful and impactful organization over the last 8 years, and it's a non-for-profit, so it's been very generously supported from Sean Parker, and as an enterprise to break down the silos and the challenges within academia and within industry, take what is really working from those two worlds and try and accelerate therapeutics through, you know, through the phase 1 space. This was the goal, and it had a short term, like the initial milestone was 5 years. Let's make an impact. So the team that Sean and Jeff built at the Parker Institute was a phenomenal team. And as I was talking with Jeff, here I'm thinking of this opportunity, that opportunity, blah, blah, blah. What do you think? It's like, why don't you come and check out the Parker Institute? But you guys don't have any labs, you know. I was looking for a research team. That's okay. It might happen down the road. Just come. So, you know, as I, of course, met his colleagues, and again, speaking to, you know, individual functions and expertise, and saw not only what was happening at the Parker Institute, but also the academic network that was involved. And so the academic network and the ideas were, in effect, the lab, at least from a discovery. That was the discovery engine. And then as far as proof of concept and generating data to support ideas, this was in the translational medicine group that we had, and this amazing bioinformatics team. And then an ability to collaborate with industry at the most promising and compelling drugs, which I started working with the Akamis Bio platform.
I see.
So here's my circle to where I am today. Because it was when I was directing research at the Parker Institute that we were looking for, okay, what's very compelling in the cell gene therapy field. And as I looked across the whole kind of field of gene therapy in the, you know, within the viral space, this platform really stood out to us in San Francisco. So I initiated a collaboration with the team at Akamis, and we generated some really cool preclinical assets. And we, you know, the Parker Institute is also funding, supporting in collaboration with Cancer Research Institute in the US, a trial within pancreatic cancer now with our lead clinical asset.
Fantastic.
So, yeah.
So that brings us back to today.
That brings us back to today.
And I think what's really interesting, Samantha, I sort of touched on it earlier, is that, you know, I've got the advantage of having you on video here. Most people will be listening to the audio, but they'll probably hear it in your voice. You're still clearly really passionate about the science, and even stuff that you did back in the early days of your career, you get excited talking about it. And I think, and at the same time, you've managed to progress your career and move into positions of responsibility and move forward in terms of your sort of own development. And I guess my question, or my my statement for you to respond to, maybe it'll be a question, I'm not sure yet, is that I think people worry at the start of their career when they're really passionate about science and they love being a scientist, that they're
Mm,
going to have to make a choice and that they're going to have to choose staying connected to the science or progressing their career. And I think, in some organizations that may be true, but I think
mm,
you've
mm,
managed to do both. Um,
mhm.
any observations on how you've done that, or what's been important to that, or kind of what you mentioned collaboration being a key thing that you've learned, but things that you've learned that have allowed you to stay connected to the science and progress your career. I know I haven't prepared you for this question.
No, that's
Apologies.
fine. I think all of the other elements of my current role, which are leadership, management, you know, now I'm a member of the executive team, of
Mm,
course, at Akamis Bio, which comes with a lot of other layers of skill sets
yes.
that one must have. All of these I still see as enabling the science is how I apply it. When you're in the PhD, you spend 90% of your time working out how to do an assay, troubleshooting,
Hmm.
and building up to the one experiment that ends up in the Nature journal.
Yes.
But that's the tip of the iceberg. All of this work has gone on underneath to achieve that key data set.
Hmm.
And in a way, I still think of that right now in my role. I do all these other things. My day is taken up with all of these other things, but it's absolutely necessary for me to sit with my scientific team and look
Yeah.
at the data. What do they need to move forward? What do we collectively think is important? And the aha moment that we have every week in our project meetings. And how do we then, I try and now and use all the skills that I've picked up through the training and development and all these wonderful opportunities I've had to get to where I am.
Mm,
Let's use those tools to continue to move it forward. And, I hope the passion and the scientific rigor is what I bring as well to my team.
right.
And I hope that I do, even though I'm not, of course, in an academic setting anymore, continue to be a scientific mentor as well,
Hmm.
to my team within the industry environment, and, always have a look on. We have kind of like journal clubs, and we're constantly doing competitive intelligence, and we're constantly looking at the field and challenging ourselves.
Yes.
And that as well for the team, for everyone in the team, it challenges everyone to be thinking outside the box and keep moving the needle.
Yeah.
Well, this is why I enjoy biotech so much, because there's no plug and play in this industry.
Mm,
So I
absolutely.
hope I answered your question.
Yeah, I think so. I think what you're saying really is that the way you affect the science progressing maybe changes, but that doesn't change the fact that you're progressing the science. Um,
Yes.
you go from doing the experiments yourself, and as you say, doing all this groundwork to get to the one that really makes a difference, to supporting other people that are doing that, but still providing the direction and providing the insight and mentorship, as you said, and I guess allowing, I guess going back to collaboration, as you said, you realized early in your career that bringing other people's perspectives in with different expertise was really critical.
Yes, absolutely.
And I suppose that has led you to then aligning all those different opinions and the areas of expertise to pursue a scientific goal and to progress the work that you're doing for the company or for the institute or for wherever you are at the time. Um,
Yeah, absolutely. And back to this just for a second, I think it's always important within a research organization to have
mm,
that space for the exploration, because no matter how smart we think we are, and no matter how many times we can draw out a model hypothesis and predict what the experiment result is going to be, we're constantly surprised.
Yes.
And, you know, having that bandwidth built into your team to be able to respond to that surprise, not ignore it because you're too busy, is also, you know, important. And something I learned back from Jeff Bluestone is don't think, you know, don't be so smart that you know the hypothesis or the rationale for this result. So he had a thing where, what's the 3 hypotheses that could explain this result?
Right.
Don't
Okay.
just think you know why you have this result. What are the alternative hypotheses and how do you test them all? I think that's what's really important, because that kind of level of diligence and thoroughness to everything that you do day to day, not taking results for granted to move things forward, you know, doing the kill experiment as you're moving a clinical candidate forward is so important, because the product that you end up, that you're putting into patients, that you're asking patients to take,
yes,
you know, especially within early stages, but in all stages of clinical development, you have to know that you've really critically analyzed that asset before it goes in. And that's something that we do a lot. Move things forward, kill experiment, have backups for if it drops dead, and make those backups even better, and just keep that pipeline rolling.
Yes, that makes total sense. And then we've probably covered most of these points, if not all of them. But if you were starting again, Samantha, if you were coming out of your PhD perhaps, or you first step into industry, or whatever it might be, or if you were talking to someone who was doing the same,
Yes.
What advice would you give them? What are the things that you think you've learned that have been most critical in your career?
I would think, you know, maybe try to go with your gut. Something that I've done a lot, you know, and as I recount my story of my career, a lot of it is around people. Oh, I met this very inspiring person, and then you know, and our jobs are very demanding. They're 24/7 for, especially within our early career, and continue to be, but you're at the bench day in, day out for years and years and years. And I think if you are in an environment that's not good for your mental health and you're not enjoying, then don't do that. That's one of my first pieces of advice that I've stuck with the whole way through, and I think is contributed to my continued excitement, because I've never had, you know, I've been so lucky not to have a kind of a toxic environment beat it out of you. Already our job is incredibly tough. You know, we're trying to figure out things that are incomprehensible and that we know small slithers of information about, and that we're interpreting wrong most of the time as we look back through the history of textbooks. Open a textbook from 10 years ago in immunology, and a lot is we think of things completely different now. So, you know, to be in a place where you can be open and yourself and allow yourself to make mistakes and have projects fail, and be able to ask the question that you think maybe isn't so smart, that a lot of times maybe is the advice that I would give to people. And I do, you know, as I speak with my mentees now.
Yeah, absolutely. I think it's critical. And then we talked a little bit about the beginning about where Akamis is and all the exciting things that are happening. What can you tell us about what's next?
Lots. Well, yes. We're heavily focused on our clinical candidate, making sure that's in the right patient population and moving that forward as we're looking for clinical proof of concept. So we're very excited about what's going to happen in the next phase for that program. We have an exciting new collaboration that my team in research and others in the company are working with within, as we already mentioned, the cell therapy space. We have lots of new potential of other kind of drug modalities also that I kind of touched on a little bit that we continue to explore and look for partners too. So it's around as we continue to build our resource bandwidth, we will continue to, as we say, put candidates through and really flex the muscles of the might of our platform.
Mm-hmm.
So lots of great work and activities coming through.
It's very exciting, and we will be keeping our fingers crossed for you. Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively so you can focus on developing medicines, treating patients, and saving lives.